Hi everyone. Today we are going to start a big discussion and start talking about the first few steps of cellular respiration. This corresponds with chapter 7 in your textbook. And we're going to take it step by step, so don't worry. It looks intimidating, but I promise it's not. So the purpose of cellular respiration is to really break down any large food molecules that we take in as food into fuel that we can actually use. So if you think about our food molecules, those are big, huge molecules. That's why we call them the macromolecules. And we need to break them down into something we can use, such as ATP. So you can kind of think of it like a meal versus a snack, or an 8-hour long sleep versus a nap. We can use the energy much more quickly when it's in smaller bite-sized pieces, aka ATP. This energy gets released when the food molecules are oxidized to carbon dioxide, which makes sense because we know from the last lecture that oxidation tends to happen when there's a lot of hydrogens, and because a lot of our macromolecules have lots of hydrogens, that is why so much energy can be released. And our focus for this will be on the oxidation of carbohydrates as I take you through the process, but cells can also use other organic molecules for energy, such as lipids, so that's important as well. When we talk about the cellular respiration cycle, the first thing that has to happen is what's known as glycolysis when we're talking about using the carbohydrate glucose. And this is not technically a step of cellular respiration in general, but it is the first thing that needs to happen in order for cellular respiration to start. So then we have what's known as pyruvate oxidation and the citric acid cycle. This is kind of where cellular respiration really begins. And then finally, we have oxidative phosphorylation, which happens through what's known as the electron transport chain. So each of these three steps is very important. And as you can see, in the end of each step, we get some ATP. And so this is really, really helpful for us because, like I said, we're converting that big food molecules into small bite-sized ATP energy. So like I said, this is just kind of a text version of what that picture was showing you. There's three major steps we're going to discuss with cellular respiration. This lecture will focus on glycolysis, but we also have the citric acid cycle as well as oxidative phosphorylation through the electron transport chain. There are two major types of reactions for the release of energy, and both of which play a role in cellular respiration. The first is known as oxidative phosphorylation, and this is similar to what we learned about yesterday. And oxidative phosphorylation accounts for almost 90% of the ATP that is produced by cellular respiration. So most of the ATP in this process gets produced through oxidative phosphorylation. This process is essentially just the transfer of inorganic phosphate, the PI, to ADP. And we are talking about the transfer of the inorganic phosphate to ADP in oxidative phosphorylation. Typically, this is how it happens. The NAD plus is reduced to NADH. The electrons get passed down that electron transport chain, and ultimately they're passed over to oxygen, which if you remember is highly electronegative, and this releases a lot of energy, which is able to be used to put that inorganic phosphate onto the ADP, thereby creating ATP. Remember, that's an endergonic reaction. The other type of reaction used for energy release is known as substrate level phosphorylation. And this is very similar, except for instead of happening through a reaction, an enzyme transfers the phosphate group from a substrate molecule to ADP, as opposed to the inorganic phosphate that gets added. So in substrate level phosphorylation, the only difference is that the phosphate group is added from a substrate molecule. This reaction still produces energy, but there is less energy released here than in oxidative phosphorylation. And this type of energy release is what you see in glycolysis and the citric acid cycle. So if you come back to this after you watch those lessons, you'll see that the most energy gets released in the electron transport chain, which is where oxidative phosphorylation happens. And we still get a little bit of energy from glycolysis and the citric acid cycle, but those make use of substrate level phosphorylation. So with that being said, now we can actually talk about glycolysis. And like I said, if we're talking about the breaking down of a carbohydrate like glucose, glycolysis needs to happen before cellular respiration can be initiated. So glyco, remember, means sugar. Lysis means to break. And so glycolysis literally means the sugar splitting reaction. And what we see in glycolysis, the main point is to break this large molecule of glucose, remember it has 1, 2, 3, 4, 5, 6 carbons, into 2 smaller sugar molecules, which we call pyruvates. And these sugar molecules each have 3 carbons. You can see it drawn here quite nicely. Glucose is, the main purpose of glycolysis is to convert, like I said, this large molecule of glucose into smaller pieces, such as pyruvate, which can be then used for the actual process of cellular respiration. Glycolysis is the one part that takes place in the cytoplasm. It actually specifically takes place in the cytosol. All the other parts of cellular respiration will happen in the mitochondria, but glycolysis happens in the cytoplasm. Another important thing to note is that this process doesn't require oxygen. That means that it's anaerobic, and that's going to be important when we talk about aerobic versus anaerobic respiration. This process does require some ATP to get the reaction started, and we get that ATP from the ATP hydrolysis we talked about previously. And also important to note about glycolysis is that no carbons are released as carbon dioxide at this stage. We talked about in overall cellular respiration how carbon dioxide gets released, but in this case all the carbons are present in the pyruvates just like we started. It has 2 major phases in glycolysis. So the first is known as the energy investment phase, and this is where the cell actually spends ATP and uses the energy stored in ATP. And the other phase is the energy payoff phase, where it actually produces more ATP as well as NADH. So the net energy we get from 1 turn of glycolysis is 2 ADP as well as 2 NADH per glucose molecule. This is a little bit of what glycolysis entails. So this is, like I said, the first step, and we get some ATP out of it. This is a really, really helpful diagram, and I'll come back to this in a little bit, but this essentially shows you the bare basics of what's going in and what's coming out of glycolysis. So you start with your glucose, you end with your pyruvates. You use 2 ATP, but what it does is produce 2 ADP. And then in the energy payoff phase, that ADP gets converted to ATP once again, but now you can see you have 4 ATP. And then your NAD plus gets reduced and becomes NADH. And in the end, what we end up seeing is a conversion to 2 pyruvate and 2 waters. Now, please note that you might be confused, oh, there's 4 ATP shown here, but remember it's about the net. So remember it took us 2 ATP to get started, so the net is only going to be 2. And then we also have our 2 NADH molecules as well. This is just, once again, another picture, very simplified version of glycolysis, but you can see the glucose is splitting. We get 2 ATP, 2 NADH, and 2 pyruvate. So stop right here in case you're freaking out, because you're probably looking at your textbook and noticing there's a lot of information in there about glycolysis. According to the College Board, you do not need to know the specific steps of glycolysis for the AP test. You don't need to know the names of the enzymes that catalyze the different steps, and you don't need to know the structure of the intermediates that form. And you're going to see this is a common trend for cellular respiration and photosynthesis. The most important things for you to know is what's coming in and what's coming out, and how is the energy being transferred throughout the cycle. That's what really matters. So this page is kind of a short version of all that you need to know about glycolysis. And when you really break it down, this is a much simpler version than what you'll probably see in your textbook. So you need to know the function, where it takes place, and remember, glycolysis takes place in a unique location compared to the rest of cellular respiration. You need to understand what's happening, what are the reactants, and what are the products. And then don't forget, once again, glycolysis is an anaerobic process. And this will be important later on when we talk about anaerobic respiration, because even if there's no oxygen present, glycolysis can still happen. So once glycolysis is finished, we have a small step known as the intermediate reactions. Because we can't actually use pyruvate to begin cellular respiration, we need to convert it into another molecule. So when we are trying to convert pyruvate, we actually do this through something known as the intermediate reactions. And this is a series of reactions that oxidizes the pyruvate and turns it into something that we can use for cellular respiration. In the presence of oxygen, pyruvate enters the mitochondria, at least in eukaryotic cells, where the oxidation of glucose gets completed. So this is the point where we move into aerobic respiration, because notice we need oxygen in order to enter the mitochondria. However, like I said, before the citric acid cycle, the next step can begin. The pyruvate must be converted into something called acetyl coenzyme A. And the way that most scientists refer to this is called acetyl-CoA. And this is the link between glycolysis and the citric acid cycle. So glycolysis gives you the 2-pyruvate, but you can't quite use that, so we convert it to acetyl-CoA. When the pyruvate is oxidized, it is converted into acetyl-CoA and carbon dioxide, which is released. And during the reaction, one NAD+ is reduced to NADH. Now, one important thing to note is, if you remember, in glycolysis, we end up with 2 pyruvates. This intermediate reaction happens 2 times. So you end up getting 2 of everything: 2 carbon dioxide, 2 NADH, and 2 acetyl-CoA. This should say NADH. This is kind of a picture of the whole thing once we get our pyruvate from glycolysis. This is one pyruvate molecule, but remember, there are actually 2 that go through this. And then pyruvate is oxidized, releasing carbon dioxide as well as NADH, and then we end up with our acetyl-CoA right here. Notice we go from 3 carbons to 2 carbons because 1 is released in the carbon dioxide. This is still following the law of conservation of mass, which is important as well. And this is just a closer up picture. So once again, in the intermediate reaction, we have a pyruvate molecule with 3 carbons, and it is oxidized and ends up releasing 1 carbon in the form of carbon dioxide, CO2. An NAD+ is converted and reduced to NADH, and then we end up with an acetyl-CoA molecule, which has 2 carbons. And then don't forget, this whole process happens 2 times, and so in the end, we end up with 2 acetyl-CoA molecules that can go through cellular respiration. So tomorrow, we will begin our next lecture, which will be on the citric acid cycle. Enjoy your day.